Hello everyone, welcome back to Study Room. So today we'll be discussing about several parts of the cell. For example, what kind of a cell is made up of nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus. So we'll be discussing about these in details. So let's start. Do you know each organ in the system performs different functions such as digestion, assimilation, and absorption because they are multicellular organisms, and we have certain categories of cells which perform certain type of functions. Similarly, different organs of a plant organism perform different functions because plants and animals we both are multicellular organism. Humans and plants, we both are multicellular organism. So that's why we have a special categories of cells to perform different functions. For example, root help in the absorption of water and minerals. Roots roots are cells. Roots have cells which absorb the water. So every cell is specialized to do a activity. You can see here we have a digestive function which is shown here. Then we have a plant's roots. So everybody perform different function. What are tissue? So cells make tissue. So tissue is a group of similar cells performing specific functions. So different cells combine and make tissue. You can see here this cell make tissue, tissue make organ, and organ makes organ system. So tissue is a group of similar cells. Now we'll be talking about basic components of the cell. So what are the basic components of the cell? The basic components of the cell are cell membrane, cytoplasm, nucleus, and plasma membrane. So this is an animal cell. You can see here plasma membrane is the outermost layer. We have Golgi apparatus. We have ribosome which stores the food. We have cytoplasm which has all the cellular organelles in it. We have lysosomes. We have mitochondria, energy providing cells. We have nucleus. Nucleus is further has inner regulatory body which is nucleolus. We have centrioles. It is only present in animal cell, not in plant cells. So centrioles are specific to animal cells. ER is smooth. ER is endoplasmic reticulum. We have RER. We have SER. SER is smooth endoplasmic reticulum. RER is rough endoplasmic reticulum. This synthesizes proteins and this synthesizes lipids. So different roles and different cell organelles for every certain role. What is cell membrane? So cell membrane separates cells from one another and also the cell from the surrounding medium. So this is cell, this is another cell. So two cells are separated by a cell membrane. It is also known as plasma membrane. So it is also called plasma membrane. The plasma membrane is porous and allows the movements of substances, material, and both inward and outward. So you can see here, this is a plasma membrane. Plasma membrane is a phospholipid structure. Phospholipid. So the golden ones are proteins. The golden ones and the blue hair, the flagella, this is lipid, and these are proteins. Which helps in the transportation of several molecules like Na and K plus channel. So cell and cell membrane, this is phospholipid, this has a phosphate head and hydrophobic tail. Tail is hydrophobic. And you can see this is also a phospholipid layer. We have membrane protein upper here. This is a 3D structure, this is a 2D structure. We have proteins here. You can see it's embedded. So this is cell membrane which separates one cell from the another and it helps in the movement of substances across the membrane. Now we have an activity observation of basic components of the plant cell. So you have to take a onion bulb, remove the dark pink covering and separate these from the fleshy white layers of the bulb with the help of forceps. Place a small piece of the thin. So you have to take a onion, you have to remove the dark pink covering, and then you have to peel a certain white color structure. You can say chilka in Hindi, and a white color chilka, a small layer, and then you have to observe it under the slide. You have to put a drop of water, you have to observe it. The thin layer can be cut into a smaller part. You can scratch it, and you have to make it smallest as possible, and observe the slide under the microscope. While observing the cover slip, so we have a So example, this is a slide. Here is your uh, this is your slide, and you have put here your material. The thing which will cover it will be called as cover slip. So this circular thing which covers it and makes a sandwich of the slide and the cover slip, and between in it is our sample. So this is cover slip. It covers it. It covers the our slide, because and it ensures that no air bubbles transfer to it, because air bubble transmission can, we can think that this is another cell. Air bubbles can be predicted as pseudo cells. So here is the video. After seeing the video, what you'll see, you can see that here are, you can see there is a bluish structure. We have used the dyes here because dyes can make us distinguish between certain types of cells. You can see hexagonal shaped cells. You can see the boundaries. Observe the boundaries here. Observe what's inside the cell. What is it moving? It is stagnant at a place. So after observing, we have conclusion here. So the boundary of the onion cell is the cell membrane, and it is covered by the another thick covering called the cell wall. So the thing which we observed here, for example, this is the cell which we saw. So the outermost layer, you can see the blackish was cell wall. This we had plasma membrane because it's a plant cell, and plant cell has cell wall and the plasma membrane. We saw the nucleus also, and the moving structure here was cytoplasm, the jelly-like substance. So this was the observation which we drew from the particular video. So this is the smallest example of how a plant cell looks like. So it has a cell wall. Cell wall is the characteristic properties of the plant cell, and even the bacteria has a cell wall. So so what, why the blue color was observed? Because we used the dyes to make it more visible and more appealing and more distinguishable. So we have been discussing about cell wall. So what is cell wall? In addition to the cell membrane, there is an outer thick layer in cells of a plant called cell wall. The additional layer which surrounds the cell membrane is required by plant for protection. So cell wall has nothing to do, but it is there for the protection of the plant cells. So you can see the plant cell here, and you can see the cell wall also here. So cell wall has a role which to protect the plants. But why do these plant cells need an extra layer of protection? Because they already have a plasma membrane just like every other organism. But why they need an extra cell wall layer, extra shield of layer? So plant cells need protection against variations in temperature. Plants have to bear certain amount of temperature. They are under constant sunlight exposure. They have to protect themselves from temperature, high wind speed, atmospheric moisture. They're exposed to these variations, certain climatic variations, which makes it important for them to have an extra shield. So that's why we need cell wall to protect it from the different climatic variations. Now we discussed about the plant cell, and the main important part was our cell wall. Now we'll be talking about animal cell. So what you have to do, take a clean toothpick, and you have, or a matchstick, or a broken something. Toothpick would be more feasible. Now you have to take the toothpick and you have to go inside your mouth, and you have to scrap inside your cheek without hurting. You can, just pinch it there and place. When you'll remove the toothpick, you have to place it under the substance that you collected, placed it under the water on a glass slide, and you have to add iodine. So you can see we are adding dyes here, and dyes here make it more distinguishable, and we can distinguish between certain cell organelles, otherwise it would be more confusing. So you have to cover it with cover slip, and then you have to add the methyl blue solution. So we are observing it under two certain type of dyes. What you'll see this. This is our observation here. So I know this is not very clear because we have used methyl blue, that's why it is blue in color. So this is the cell which you'll see. This is a cell. The three things which is common in every cell is a cell membrane, nucleus, and a cytoplasm. So cell membrane is in every animal cell, nucleus and cytoplasm. We notice several cells in the scrapped material. There are cell membrane, cytoplasm, and nucleus. So you have to observe these three things in animal cell. Cell membrane, nucleus, cytoplasm. In plant cell, you have to observe cell wall, cell membrane if possible, cytoplasm, and the nucleus. So this is the major difference. What is cytoplasm? We have been discussing about cytoplasm. It is the jelly-like substance present between the cell membrane and the nucleus. So this is nucleus, this is cell membrane, and the yellow part here is cytoplasm. Cytoplasm is has various several cell organelles of different varieties. There are mitochondria, Golgi bodies, ribosome, which floats in the cytoplasm. Nucleus. It is an important component of the living cell. It is generally spherical and it is the guiding center of the cell. It regulates everything. It can be stained and easily visible under the microscope. So when you were observing the plant cell and animal cell, the only prominent structure which we were able to see is nucleus. It was very dark. It could be seen very easily. It can be located very easily. And nucleus is separated from the cytoplasm by a layer called nuclear membrane. So nucleus have its own membrane which we call as nuclear membrane. You can see here, this is the nucleus. So nucleus is circular in structure, and we have cut it. So this is the outermost layer is the our nuclear membrane. Then we have nuclear envelope inside. Inside the nucleus we have nucleolus, and we have chromatin. Chromatin contains the genetic information. And we have certain pores over our nuclear envelope because DNA has to transfer the information outside of our bodies so that's why it has to emerge out of the nucleus to pass it from different cells to another. So it's important that it has certain pores over it. So this was about nucleus. Now we'll be observing a video.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material.
So in this video, what we can see is, try to see what it is. It is a cell. Cell is the basic and smallest unit of our organisms, and every cell have three things in common. So it has a cell membrane which differentiates the cell from the outer environment. It protects the cell from the outer environment. We have cytoplasm, which is the jelly-like fluid. Then we have the DNA. DNA is the cell's genetic material. So these three things are common in every cell. So I hope now the cell part is clear. The basic components of the cell is clear. Let's move ahead now. So in this video, we basically got to know what are the different components of our cells. It has a nucleus. Nucleus is the guiding center of our cells. It has DNA, which encodes the genetic information. Then we talked about cytoplasm. Cytoplasm has several cell organelles in it. For example, Golgi apparatus, ER, ribosomes. Then we talked about cell wall. Cell wall is there for the protection of plant cell. Basically, it is not present in the animal cells. So these were certain parts of the cell. In the coming video, we'll be talking about Golgi apparatus, ribosomes, mitochondria. So it's all going to be very, very interesting. So I'll see you in the next video. Thank you.